March 24, paper 6. The same ideas that came in all the previous exams were here. Same ideas. Question 1. A student investigated the effect of glucose concentration on the rate of anaerobic respiration of yeast. I'm investigating the effect of glucose concentration. يبقى the word after effect of is my independent variable. This is what I'm going to change. I'm going to change the glucose concentration and see how it affects the anaerobic respiration. What does anaerobic respiration do? The yeast breaks down the glucose into ethanol and carbon dioxide. In a previous exam, he got you the bread dough, and he put yeast in it. And we measured the change in height of dough. But in this exam, what is he measuring? He gave you a hint here. Anaerobic respiration causes the blue dye methylene blue to become colorless. It causes a change in color from blue to colorless. The time taken for the blue color to disappear is used as a measure of the rate of respiration. يبقى this is my dependent variable here. This is my dependent variable. This is my results in this experiment. Okay? The student used this method. Label one test tube 0.0%, 0.5%, and 1%. These are the three glucose concentrations I'm going to use. Put 5 cubic centimeter of water into the test tube 0. I only put water here because I'm not putting any glucose, just water in this test tube. In the second test tube, I'm going to put 2.5 cubic centimeter of the glucose solution and 2.5 cubic centimeter of water. يبقى half glucose, half water, so I can get this concentration. In the third test tube, I'm going to put only glucose solution, 5 cubic centimeter of 1% glucose solution. These are the three concentrations I prepared. 3 concentrations. Okay. Stir the contents containing the yeast. I highlighted this. So, of course, there will be a question on the stirring part, and we answered it before. Every time he asks you about the stirring, why do I stir the beaker? Why do I stir inside the beaker? Because for even distribution, I want the yeast cells and the glucose to be evenly distributed in the cup or in the test tube to react together. Okay, add 5 cubic centimeter of yeast suspension to each test tubes. In all the test tubes, I added the same volume of yeast. Okay, this is a constant variable. Same volume of yeast in all, this is a constant variable. Put all 3 test tubes into a water bath, 40 degrees Celsius. I put them all in the same temperature. However, this is an error because the temperature is not maintained. It will go colder. Start the stop clock. Wait for 3 minutes. Before starting to measure the time, I waited for 3 minutes before adding the methylene blue. Why do I wait? I put them in the water bath and I wait. We answered this before. It is for equilibration. Equilibration. What does the word equilibration mean? It means that the test tube will reach the same temperature as the water. After 3 minutes, remove the test tubes from the water bath. Place them in a rack. Add 1 drop of methylene blue to each test tube and carefully mix the contents. Use a second pipette to add a layer of oil. Why do we add a layer of oil on top of the yeast? We know that the yeast carries out anaerobic respiration. So you have to put oil to prevent the oxygen from going in. يبقى this oil he wrote it already to maintain anaerobic conditions أو prevent oxygen so that the yeast can do anaerobic respiration. Okay. Put the test tubes back in the water bath. Start the stop clock. Measure the time taken for the blue color to disappear. There is an error here. Oh, actually, there are two errors, two sources of errors. The first error, we said before that we cannot do all the test tubes at once because you will not be able to concentrate with the 3 test tubes at the same time. What is better? Do each test tube separately. The second error, anything with color is difficult to judge by your eye. So we are supposed to use a colorimeter. Record the time taken in seconds for each test tube. The students stopped the time if the blue color had not disappeared after 10 minutes, and they recorded the result as more than 600. طيب, he gave me the results. These are the results for each test tube. This one, until 10 minutes, it did not change. يبقى I will write more than 600. This took 600, 6 minutes, 2 seconds, 2 minutes, 12 seconds. Prepare a table. Of course, he wants you to convert the minutes to seconds. I told you before that. How do I do the table? Changed variable is on the left, results on the right. What is my changed variable? The glucose concentration. Percentage concentration of glucose. What are my results? The time taken for the blue color to disappear. يبقى this is how I should do the table. Glucose concentration / percentage. These are my concentrations. Time for the glucose to disappear. The first one took more than 10 minutes, so I wrote it more than 600. Here I converted it to seconds, 362 and 132. This is my table. State a conclusion. What is my conclusion? The higher the glucose concentration, the less time taken to disappear, which means that faster anaerobic respiration. The greater the glucose concentration, the higher the respiration rate. What is my independent variable? Independent هو changed. What did I change? I changed the glucose concentration. What is the variable that is kept constant? I can say anything from the experiment. For example, the volume of yeast or the temperature, they were constant. Explain why it is important to stir the yeast. We just said it. Stirring is important for even distribution of the yeast cells and the glucose in order to react together. One way to improve this investigation would be to use an increased number of different concentrations of glucose. Suggest 2 other ways to improve the investigation. I need 2 improvements. As I just said, look for the errors. What are the errors we said? We said that he did the test tubes at the same time. We said that the color, we're judging the color by our eyes. Okay? Therefore, you can say, do each concentration separately. Use a colorimeter. Repeat each concentration. Okay, more than once and calculate average. Describe how you could make 5 cubic centimeter of 0.25 glucose solution using 0.50 glucose solution and distilled water. Fine. Let's think. If you have a glucose solution that the concentration of this glucose solution is 0.50. Uh, you want you want to do you want to prepare your own concentration. You want to prepare a test tube with 0.525%. I want to prepare 0.25% from this one. طب what is the volume? 5 cubic centimeter, 0.25%. This is what you want to prepare from this one. So what are you going to do? I am going to take 2.5 cubic centimeter from this solution plus 2.5 cubic centimeter water. So you will divide the concentration in half. How did I divide the concentration in half? Because I added half water, half glucose solution. يبقى again, if you want to prepare 5 cubic centimeter, 0.25% glucose from a solution that is 0.50 glucose, what are you going to do? You're going to take half water, half glucose solution, 2.5 cubic centimeter of the 0.50 glucose solution, 2.5 cubic centimeter of water. Describe the method you test a solution for the presence of glucose. Easy question. Add Benedict reagent, heat in a water bath. A student investigated the effect of temperature on the rate of respiration in yeast at 25 degrees Celsius and 35 degrees Celsius. This is another way. I am going to investigate effect of temperature. This is my independent variable. And I'm going to check the rate of respiration at these two temperatures, 25 degrees, 35 degrees. How am I going to measure my results? What is my dependent variable? Dependent. I'm going to measure the volume of carbon dioxide produced by the yeast every 5 minutes for 30 minutes. This is my dependent variable here. طب he asked you, how are you going to collect and measure the volume of carbon dioxide? Just one mark. I can tell him using a gas syringe or using a delivery tube underwater and an inverted measuring cylinder. This delivery tube underwater and an inverted measuring cylinder with graduations. You can use either this one or this one. The student did 3 experiments at each temperature. Take a look at this graph. They used the results to calculate the mean volume. Take a look at the table, sorry. Calculate the mean volume of carbon dioxide. At each temperature, he gave me the table for temperature 35 degrees. After 15 minutes, he counted the volume 3 times, okay? And he calculated the mean. The student decided that the results for one of the experiments is anomalous. We just solved this in another exam. What is the meaning of anomalous result? Anomalous means that it does not fit the pattern. Look here. This is 1.8 and this is 2. This is 3.2. It is very far from the other two numbers, so it is probably wrong. This is an anomalous result. What is an anomalous result? Result that does not fit the pattern. It does not fit the pattern. Okay, and then he asks you again, how does the student calculate the mean volume of carbon dioxide produced? Not in general, يعني. In general, how do I calculate the mean? I add all the numbers over over the number of data you have. But he's not asking you in general. He's asking you specifically in this experiment. In this table. Since you have an anomalous result, you do not include it in the average. How am I going to calculate the average here? I will not include sample 2 in the calculation because it is anomalous. The results of the whole investigation about the effect of temperature on the rate of respiration is shown. This is the table. Take a look at it, kid. When you measure the results every 5 minutes, he puts the time on the left side, like we just said. And I have the mean volume of carbon dioxide at 25 degrees, mean volume at 35 degrees. These are your results. Using the data, compare the mean volumes at 25 and 35. Compare. What is a comparison that you can see? For both of them, the volumes increase with time. Of course, at 35 degrees, the numbers are always higher than at 25 degrees Celsius. At 25 degrees Celsius, it continues to increase, while at 35 degrees Celsius, it increases and then it levels off. It becomes constant. This is how you compare. Any comparison, any two comparisons between the two sets of data. Volume of carbon dioxide is always higher at 35 degrees Celsius. At 25 degrees Celsius, volume of carbon dioxide continues to increase, while at 35, it levels off. It means that it becomes constant at the end. Okay. Plot a line graph of the mean volume of carbon dioxide using all the data. I want to plot a line graph for all of this. What do I do? Left side of the table is on the x-axis. This is on the x-axis. Right side on the y-axis, but I'm going to plot two lines. On the x-axis, I will put my scale 5, 10, 15, 20, 25, 30. طيب بس look. I'm trying to put the scale as large as possible in order to fit my numbers. So what did I do? I started here, 0, 10, 20, 30. This is how it will fit. On the y-axis, what are your numbers? The smallest number is 0. The biggest number is 3.2. 0 لحد 3.2. So you want to put a scale to cover all this. Say I will put from 0 to 4. 0 to 4. 0 to 4 on the y-axis. But look how many boxes you have. You have to cover all these boxes. So you have to put a small interval. How did I put it? 0, 0.5. 1, 1.5. 2, 2.5. 3, 3.5. 4. Okay? And then I'm going to plot my points for each line. For the first line, at 5 minutes, it was 0. 5 minutes, it was 0. 10 minutes, it was 0. At 15 minutes is in the middle between 10 and 20. 2 boxes, it is 0.1. Every 2 boxes on the y-axis is 0.1. And then 0.4, and then 0.2, sorry, 0.25. I'll plot all the numbers. 0.2, 0.5, 0.1. Okay? This is 0.5. Let's correct the line. Okay? Okay, now it's correct. 0.5, and I put my line, draw the line point to point. For the other graph, I'll also plot my points, but I will not put them as x's. I will put them as a dot and a circle. They should be different. Okay? 0.1, 0.8, 1.9. I will plot my points, and then I will draw the line point to point using a ruler. Don't forget to put the key to differentiate between the two graphs. This is the 25 degrees Celsius, and this is the 35 degrees Celsius. Okay? Estimate the time taken to produce 3 cubic centimeter of carbon dioxide at 35 degrees Celsius. You have to show him. You will draw a line from the 3 cubic centimeter until you meet your graph. I want it the 35 degrees Celsius. So until I meet this graph, and then I go down, 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 down. It will be 23 minutes. 23 minutes is the time at which it will produce 3 cubic centimeter. Carbon dioxide was bubbled through the hydrogen carbonate indicator. The hydrogen carbonate indicator, we took it before. It is red before the gas was bubbled through. When the carbon dioxide bubbles, reacts with the hydrogen carbonate, it will give me yellow color. Okay, this is the first question. 27 marks for the first question. Let's check the second question, which is much easier. This is the cells from the red onion. He wants you to draw cell X. I will draw the same shape of the cell. Same shape of the cell. Okay, it's not rectangular. It has like a point from here. Okay, and here. So I have to draw it the same shape. And the vacuole with points, extensions, and the nucleus. Please do not color anything. I drew the cell wall as double lines because it is thick. I can see it as a thick line. And you have to draw these extensions of the cytoplasm and the nucleus without coloring anything. After the drawing, we have the magnification. Measure the line of PQ. Measure the line PQ. I measured it using my ruler. It is 78 millimeter. Calculate the actual length. I want the actual length. Draw the triangle. Magnification equals image over actual. I want the actual, therefore image over magnification. Where is my magnification? Under the picture. 78 over 340. Okay. I want the answer to 3 significant figures, so I will round it up. Here is my 3 significant figures. Water moves in and out of the cells by osmosis. Plan an investigation to determine the effect of concentration of sodium chloride on osmosis. This is an experiment about osmosis. I'm going to change the concentration of sodium chloride and see what happens in osmosis in the plant tissue. This is an experiment we took in our notes when we studied the osmosis chapter. You can bring any plant tissue, potato, onions, whatever. What are you going to change? I'm going to change the concentration of sodium chloride. Concentration of sodium chloride. Okay? يبقى put each in a different concentration of sodium chloride. And then, what am I going to do? Constant variables, as much as you can, at least 3 points. Same type of plant tissue, same mass, same initial mass of plant tissue, potatoes مثلا, same temperature, same volume of sodium chloride solution. Everything should be the same. I'm only changing the concentration of sodium chloride. Okay. And then, what are you going to measure at the end? What are your results? Measure the final mass. You measure the initial mass, leave the potatoes in the sodium chloride solution for 10 minutes, and then measure the final mass. Calculate the change in mass. Use thermostatically controlled water bath to maintain the temperature. Use a cutting board while cutting the potatoes. This is my safety precaution. Repeat at least 2 times and calculate an average.